morning. My name is Selena Ward, and today I will be discussing a paper entitled A Marked Increase in Community-Associated Methicillin-Resistant Staphylococcus aureus Infections in Western Australia between 2004 and 2018. This paper was written by 4 authors: Dr. Bloom, Professor Coombs, Ms. Tampone, and Dr. Armstrong, all of which currently work for respectable teaching universities and associated laboratories here in Western Australia. This article was published in Epidemiology and Infection, which is a subset of the Cambridge Press. The impact factor of a journal is a measure of the frequency in which the average article is cited within a particular year. For this article journal in particular, the impact factor is 2.075, which is good but not great. The peer review process for this journal is that of a single-blinded independent expert referee. Overall, this presentation has a goal of critically appraising this article, and I will then move on to explore how the epidemiology data can then be used to support the implementation of infection control guidelines and practices. A bit of background information. Statewide policy management for MRSA in Western Australia has now grown to currently include surveillance swabs for all patients and healthcare workers before entering Western Australia, myself included, and statutory notification of case detection and with follow-up by the relevant public health units and communicable disease centre. Western Australia has also adopted rapid laboratory testing for MRSA in order to detect the clone genetics using multi-locus sequence typing. This then allows to determine the genetic traits and classification into PVL positive and PVL negative. PVL stands for Panton-Valentine leukocidin, which is a virulence factor that causes tissue necrosis and leukocyte death. Figure 1 highlights the differences between hospital-acquired and community-acquired MRSA strains. As you can see here, hospital-acquired strains are generally PVL negative, whereas those of community-associated strains have a bit of a variance between PVL positive and PVL negative. These stringent MRSA policy actions have allowed Western Australia to continually have the lowest rate of MRSA occurrence in Australia. This has been described back in 2011 in a paper by Coombs et al. As we can see here, the figures for Western Australia of MRSA in comparison to Staphylococcus aureus is that of 66 within a total of 332. We can then compare this information to every other state and see that Western Australia has a much lower percentage of MRSA compared to every other state. The important thing to consider here is whether or not this epidemiology data can still be supported if this sort of a table were reproduced now in 2021. From the article, the study design follows a retrospective ecological study. The overall aim was to describe the changing epidemiology of community-associated methicillin-resistant Staphylococcus aureus specimens in Western Australia between 2004 and 2018. For timing's sake, here on forward, I will be referring to community-associated methicillin-resistant Staphylococcus aureus as CA-MRSA. In the study, the independent variables that we were considered are that of geographic region classed according to climactic zones, as we can see here in figure 3, the year of specimen collection, and the age and gender of individuals that the specimen was collected from. The dependent variables of interest were that of the incidence of MRSA occurrence and the proportion of isolates that carry the PVL genes. This is important in order to survey the emergence of genetic clones of MRSA in order to assist and determine the appropriate antimicrobial prescribing, policy recommendations, and practices for guiding patient-centered care. Methodology. For this article, they had a pretty strict inclusion and exclusion criteria. As we can see, samples were collected from individuals with a Western Australia address. Duplicate samples were excluded. The samples positive for CA-MRSA must have been verified via MLST sequencing, and we can also see that screening specimens were excluded. In saying this, the exclusion of screening specimens, it means that pretty much all of our samples were collected from individuals with an infection rather than colonization. From a genotyping perspective, the process used was that of PCR amplification of the DNA of the MRSA, which is then used for the MLST sequencing. The statistical analysis performed in this paper was completed using the R software. They calculated the annual incidence rate compared to the population that was taken from the Australian Bureau of Statistics. The Wilcoxon sum rank test was calculated, as well as multivariate logistic regression and negative binomial regression. As we can see here from the overall findings and good graphical presentation of the incidence rates, we can see that for all 4 climactic zones, as classified by above, we can see an increase in the red, PVL positive, and a slight increase in the PVL negative. What's important to note here is the overall pattern within all 4 climactic zones. We have an ongoing trend upwards. What I think could have been done better here was to not stratify all these into the separate climate zones, but also have a comparison of the overall Western Australia trend of PVL positive and PVL negative. In saying that, I'm sure that it would follow a similar trend with a quite steep uprise in the PVL positive and a steady rise in the PVL negative cases. Major findings from this summary. The most important thing to take away from this is that there was a total of 5,700,000 cases of CR-MRSA in 2004 to 2018. Of this number, 57.6% were PVL negative and 42.4% were PVL positive. From this, the proportion of PVL positive has increased from a very low 3.4% to 59.8%, which is explained in this very high upward trend here. The median age, though, of these PVL cases was significantly lower at 26.7 years, whereas for those with PVL negative cases, the median age was 54.5. This is a pretty important thing to consider when you think about the migration pattern. I personally, from living in WA, know that we have a lot of young backpackers and international tourists. Could this potentially be a contributing factor to the rise in the PVL positive MRSA cases in a younger population? We don't entirely know. The findings relevant to clinical practice is mostly the annual incidence rates of CA-MRSA, in that has increased almost a 3-fold rate from 86.2 per 100,000 of the population to 245 per 100 of the population. As we can see here, we have the stratified incidence rates ratios, and for all isolates, it's quite a big number. The emergence of the 2 PVL positive clones, being that of the Queensland CA-MRSA and the WA CA-MRSA, account for the majority of cases within this study period. From a proportional perspective, Queensland CA-MRSA cases accounted for nearly 1.6% of all of CA-MRSA in 2004, but this then, however, increased to 37.9% of the total by 2018. Again, a big jump. From a critical analysis perspective, there was a few limitations identified within this study. Firstly, the inability to account for changes in environmental and healthcare workers' behaviour over time. How this would be done, I'm personally not entirely sure, but it would definitely involve a large amount of confounding or association modelling. Secondly, there is no comparison with the population changes or migration patterns over time. Thirdly, WA often has a lot of FIFO and interstate travellers coming through, a lot shared with the Northern Territory and with Southern Australia. Could this potentially be a confounding factor that increases the CA-MRSA rates and emergence of new strains? It would be interesting to know. Lastly, there is no information on the corresponding antimicrobial sensitivities of these MRSA strains. All we know is that they have resistance to methicillin-based antimicrobials. From a statistical point of view, we can overall confirm that there is statistical validation. The incidence rate ratio is less than 1. The 95% confidence interval is narrow, and the incidence rate ratio is within that 95% confidence interval. From a study design point of view, we have meeting criterion over 4 areas. Firstly is that of construct, and the ecological studies are quite useful for studying the epidemiology of bacteria and determining these patterns and trends. From a content point of view, we know that negative binomial regression modelling and multivariate logistic regression modelling are good measures for incidence and association. From a face criterion perspective, the contents of this paper are relevant to what they were trying to achieve. Lastly, the most important part, from a criterion perspective, the results from this study are then supported by similar studies. As we can see here, there are a few examples from similar timeframes across Australia. Overall, the surveillance and emergence of genetic clones of MRSA emergence assists us to determine the appropriate antimicrobial prescribing, policy recommendations for practice, and is useful for guiding patient-centred care. In applying to the wider population, we can see that this demonstrates an increasing rate of emerging antibiotic resistance infections, which then again comes back to highlighting the importance of adhering to strict infection control measures. From a nursing perspective, this will help to enhance knowledge and understanding of the clinical processes to monitor progression of likely disease patterns. Importantly, from a medical perspective, this will also help to influence prescribing patterns by knowing the likely organisms for each population and region. Again, referring back to our map, we now know and have a bit of an estimation of the likely emergence patterns of PVL positive and PVL negative for those areas. Finally, from a management perspective, this is important for policy decisions regarding the use of precautions and screening for both patients and healthcare workers. In conclusion, how can this be used to inform practice? Firstly, the surveillance and emergence of genetic clones of MRSA emergence assists to determine appropriate antimicrobial prescribing, policy recommendations for practice, and guides patient-centred care. The findings from this study overall do show that there is an increase in the proportion of CA-MRSA within the Western Australian district. An important thing to consider in taking away from this is for patients presenting with skin infections, it's very important to consider the possibility of MRSA as opposed to a standard infection and adopt the appropriate precautions as required. Personally, I am hoping to go into research about multi-resistant organisms, and the knowledge that I have gained from this presentation and analysis will assist me to build an understanding of epidemiology and the rise of CA-MRSA. Thank you for listening. Here is a list of my references.